Insights that we've been having from a disease model that have had kind of profound basic implications, particularly for how the central nervous system controls reproduction and puberty. Now, for an audience like this, it's a quick introduction to the holy trinity of the axis. It's in the name of the hypothalamus and of the pituitary and of the gonads, whether it's an ovary or a testes, and of the releasing factor and tropic hormones which drive it, and the sex steroids and peptides that feed back. With that introduction, we can now focus on the king of this whole process, which is the Gnrh neurons that govern this whole process. To study Gnrh in the human, if you wish to choose that as your sort of limitation, there's a daunting methodologic problem, and there's only 1,200 of these neurons in the entire brain. They're very small and widely dispersed. They're problematic for endocrinologists because you can't measure Gnrh, and in fact, you have to measure secondary secretagogues such as LH and free alpha subunit, and you have to do so frequently at 10-minute intervals. These are daunting challenges, and when you add the fact that a lot of this research has to happen in children and at puberty, you really have some formidable obstacles. So at this point, the young people in the audience are wondering, why should I ever get involved in something that's this complicated? Well, actually, the rewards are pretty significant, because Gnrh and kisspeptin, our neurons, are the master regulator of all the reproductive axis. They really are the pilot lights of reproduction. But more importantly, for an individual, this system is critical for evolution of the species. This is what determines reproductive fitness to the environment, and it must, over evolution, have adapted to nutrition, olfaction, exercise, predator density, to exert fertility only in the time of choosing for optimum species. It's the one hormone and system that is not unique to the individual, but is given the responsibility of the species. Remember that fact, because in a moment we're going to come up to some surprises that actually changed my whole career and our whole way of doing business. It's also, if you think about the hypothalamus and the species mandates that it gives in the brain, the first is to really grow and develop, and that's the growth hormone axis and to some extent insulin. The second is to deal with stress, which is the other component of this meeting. And the third one really is to go forth and multiply, which is the species mandate, not the individual mandate. So here's the surprise. The surprise is that when the human genome, rough draft, was developed in 1990s, for all of this power that the reproductive system has, there was only one gene coding for this in the entire human genome. That was astonishing because the redundancy characteristics of GPCRs, transcription factors, second messengers, and requiring biologic backup, here was the one system that had none. Secondly, it was discordant with a number of clinical and genetic findings, recessive dominant, X-linked inheritance of these patients, and the complex developmental biology that has to underlie it. So to us, this was a demand bid for changing direction completely. We went from being essentially physiologists and doing Gnrh agonist studies, as was alluded to in the introduction, with Wiley and John Riviere for 16 years in precocious puberty. That was the foundation for the $3.2 billion market of Gnrh agonists. It was the proof of principle in the human. And we redirected our entire research program to genetics beginning in 1996. And what I'm going to talk to you about today is a result of that. Now, it does help when Science magazine like this points up in its 2005 issue, the 125th year of science, looked at the 125 great unanswered questions, and number 84 was what causes childhood to end, and of course that's puberty. The other thing for me as a clinical investigator is I saw this patient in the clinic. He had first been seen in 1943 by Fuller Albright. And he came in because of a complete absence of puberty. At age 28, he, as you can see, had a small phallus, a female escutcheon, small testes, and could not smell. So even a quick look at a fellow like this will tell you that he's got a reproductive problem. At the time, Albright had severe Parkinson's disease, and he could no longer write in the chart. So he was always assigned fellows who were, take down what Albright said, And the two fellows who saw this patient with him, Dr. Kleinfelter and Dr. Reifenstein, the fellows at the time, Albright said to them, if we took urinary gonadotropins, these new measuring sticks of pituitary output, I think what we're going to find is that most people have normal urinary gonadotropins. Kleinfelter discovered that there was a group of men with elevated gonadotropins that looked just like postmenopausal women. But this patient had a low level of gonadotropins. They obtained a skull film. He didn't have a pituitary tumor. And he was listed as isolated gonadotropin deficiency. This was what they were carried in OMIM, online Mendelian inheritance in man, for many years. Now, by the time I was a fellow in 1977, I had seen this patient, and I went to a conference in which Wiley Vale was sitting next to me, and we went and had lunch with he and John Riviere, and we couldn't get the peptide, either the agonist or the GnRH itself, because my hypothesis was that this wasn't really a pituitary defect at all, but that this was a hypothalamic defect. And we didn't have the term knockout at the time, but that was what we were thinking about. This is where Wiley became very important. Here's a good example of what it looks like when you study a normal male patient at 10-minute intervals, because you have to draw bloods at LH at 10-minute intervals. You can see quite clearly the pulses of each LH that is inferred and actually proven to be following a GnRH pulse. And you can see the testosterone up there of a normal male. Here's the patient I just showed you. This patient clearly has no gonadotropins, no pulses, etc. And then using Wiley and John's GnRH, we found out that in collaboration with Dean Kamen, who made pumps, we could give this in a pulsatile fashion. And you'll notice that when we did that, we were rewarded with pulsatile LH. See the different colors there? Each one of those is a different dose. So not only could we change this patient into a fully normal reproductive male, which we reported in the New England Journal as a therapy, we can also construct log-logit curves. This is a human model here, that you're doing dose-related pharmacology here in a pulsatile system. And that set the base for 20 years of physiology. Now, at the same time, this experiment was done by the Rockefeller Institute, Donald Pfaff, who asked a very simple, blindingly clear question: where does GnRH neurons come from? Did immunocytochemistry, and was astonished to find out they weren't in the brain. They actually were extramural to the central nervous system. And see where the olfactory probe is? They start in the olfactory placode. They migrate in by day 14. And by day 16, they have taken up residence in the hypothalamus, exiting from whatever migratory pathway they have. So this is a pretty complicated developmental biology for GnRH neurons. And the first patient, they, by the way, go from being migratory neurons that have a lot of properties of migratory neurons to more mature neurons that are secrete GnRH by the end of this. This baby was then discovered in the same year that had many complicated genetic syndromes. Kalman Syndrome, which we'll talk about, that's the anosmia that our patient had with low gonadotropins, ichthyosis, chondrodysplasia punctata. And during the neonatal window, when the gonadotropins should be up, the gonadotropins were low, testosterone was low, and confirmed to have Kalman syndrome. The baby died, and this is the olfactory placode, showing no olfactory bulbs or tracts. And it turns out that, as you can see in the upper region, his mother lacked tip of an X chromosome, which she gave to him. And all of these syndromes were X-linked, and so the tip of the X chromosome. So it was a structural genetic defect that turned up the defect that discovered where the Cal gene was. Now, it turns out that this woman eventually had a miscarriage and conceived a second time, had the same immunocytochemistry, and therefore underwent a therapeutic termination at 19 weeks. A normal fetus would actually have, as you can see here, these are GnRH neurons by gold staining in the medial basal hypothalamus by 19 weeks. This whole process is done. But the Cal fetus at 19 weeks had no GnRH neurons. But in fact, the GnRH neurons were in the nasal septum, and they were migrating in till they got to the cribriform plate. And see that gnarled traffic jam there that sort of looks like the beginning of the polio. In fact, those are the GnRH neurons all waiting for the last one to join. It takes about an hour and a half, as best I can tell. So as a result, there winds up being genes that control this stem cell biology of the olfactory placode the migration through the cribriform plate, and then eventually into a long-distance migration where these Gnrh neurons come to the hypothalamus, take up residence, and control the pituitary. Given this as a background, if you're a clinician and clinical investigator, you see this disease, which is now called isolated Gnrh deficiency. And in fact, it's extremely rare disease. It's 1 in 30,000 for males and 1 in 120,000 for females. Because of our interest in this and because we had pulsatile Gnrh therapy, we've been fortunate enough to accumulate 1,800 cases of this and have DNA on these cases over the years. It's a 30-year piece of work. And they divide themselves up into 2 groups. The group that does have olfactory bulb abnormalities and anosmia, and they're called Kalman Syndrome. 1944, the year after Albright described this case, Kalman described his case. And as you'll see in a moment, all these genes and pathways, because I'm going to talk a little about the bioinformatic pathways here, have a neurodevelopmental theme. The normosmic variant, which is about 50% or so, that have normal olfactory bulbs and tracts, these are genes which turn out to be neuroendocrine control. And this human disease model has been quite prismatic in showing us these genes. Now, we had a fellow who came from Kuwait. And since the Cal gene had been described, I sent him back and said, find me a normosmic family with isolated Gnrh deficiency. Gave him one of Dean Kamen's pumps and sent him back. So he goes on television in Kuwait, 800 channels on direct TV, what's on tonight, ovulation induction, and he finds this family, which comes forth, and very typical, and why did I say Bedouins, that you have, in fact, 2 brothers and a sister that marry their first cousins, and in fact gives rise to 8 of these cases. Now this is a region that's 80% of southern Saudi Arabia marry second cousins or closer. It's characteristic of this, because remember, they're Bedouins, and they migrate in small groups. It has the advantage of a close nuclear family. Your uncle becomes your father-in-law, and no dowry is needed. So for poor migrating populations, this is the preferred way of breeding, as opposed to, say, Japan, where they always bring in a commoner to marry into royal stock. This is an inbreeding. It's also characteristic. Jewish families used to intermarry this way. Cohens were to marry Cohens. And so it's a characteristic of the Middle East, and it gives rise to your ability to do linkage studies by homozygosity mapping. We took this family, we localized this very quickly to the 8 chromosome. We developed a series of candidate receptors and developed that this new receptor that had not been identified before was a complete loss of function due to a terminating mutation. Now, at the same time, Steve O'Reilly, who was a friend of mine when he was in Boston, actually, you may remember him, he went back and, with a company in England, had been knocking out the GPCR family of receptors with a technology that was interesting, and they would go and knock out the rhodopsin receptor family, the druggable family. But before they did that, they would go to an orphan website, and they would look up a name, and they'd give the, I mean, GPCR 54 is not a very sexy name, actually. If you're in a pharmaceutical company, you got to give it something better. So it was called Harry Potter, was the mutation that they created. Now, I explained carefully to Steve, who's not stupid, I said, this is the wrong phenotype. You see, the phenotype is that you don't go through puberty, so it should have been Peter Pan was the orphan of the receptor, but it turned out that, in fact, this turns out to be the same receptor. This was published in the New England Journal. It's a double-length article, and the first time the New England Journal published an article with human and animal data in it. And to give you a good example of how empowering human genetic research can be, it was done by Stephanie Seminara, who was a fellow going on the faculty at that point. She wound up being the first author on this. Look at what happens when an observation is made that you already know is in a human. These are the number of papers that followed that. 99% of these are in basic science, because of the implication of having been directed by human genetics to a target that no one knew anything about. So that's kind of the theme today. And in fact, this diagram I showed you is now getting much more complicated, because not only is the KISS receptor and ligand here, it turns out that the neurokinin B and dynorphin neurons also are in these, and they're called candy neurons. So there's a whole layer of neuronal things in all mammalian systems that characterize this and control it. Knockouts of either one of these, tachykinin or neurokinin, its other nom de plume, create hypogonadotropic hypogonadism. Here's FGFR1. This is one of the receptors that I was talking to Ron about last night. This is one of the receptors that causes Gnrh through another chromosomal break. It turns out it has 3 IgG domains. These 2 yellow ones are the ligand binding pocket. And when we and others looked, there are a series of point mutations here all the way up and down this receptor transmembrane domain and protein tyrosine kinase domain. See this red mutation here in ligand binding pocket number 2? Remember that because I'm going to show you this is where clothal binds as a cofactor for FGFR1. But in fact, when we look at it, having the luxury of both, we could see that this also causes normosmic IHH. Now remember, I'd sort of set up a dichotomy here, that one relates to migrating neurons, one relates to both. And this was our first hint that some genes played a role in both of these pathways. This mutation is something that when you look at where it is and look at X-ray crystallography, this is the gene I just showed you. This is the blow-up of the X-ray crystallography of the 2 ligand binding domains. And down here in the 3rd pocket is a leucine to serine mutation here. That's important for 2 reasons. It's deep in the ligand binding pocket. And one of the questions is, how are you going to sort out the 23 FGFR1s with 4 receptors, which binds to which? Well, length is one of these things. Assuming a patient has Kalman syndrome, they have to have a ligand mutation, if they have one, that reaches this point. If you line them up, there's only 3 long enough to reach this area, which in fact is FGF8, which we discovered and had mutations. But here's the biologic proof of that. This is a bioreactor, and when you look at this, this is FGFR1, the wild-type receptor with FGF1. This is the same wild-type receptor with FGF2 and with FGF8. So FGF doesn't distinguish the binding of any one of those 3. If you take the mutant receptor, FGFR1 binds to it just fine, FGF2 binds to it, but it completely eliminates the binding of FGF8. And in fact, we screened FGF8 and found a series of mutations. Not only did we find those, and by the way, this is the clothal binding site, so this has actually got a lot of importance because of the cofactors, and nutrition bumps up against FGFR1 via clothal beta, by the way. But in addition, if you look at the SYN expression group of the genes that are expressed here, you can see that here's Cal gene, here's FGFR1, here's FGF8, and here's a few other genes which we went through and developed mutations, interleukin receptor number 17, DUSP16, Esprouti, a whole bunch of these, which, by the way, you would never pick up if you're trying to pick up a candidate gene and try it in a disease, but now genetics tells you these are all involved. Not only that, it also tells you that most of these have Kalman syndrome, each one of these genes, and it's really FGFR1 that seems to play in both the neuronal pathways in the hypothalamus and development. But this is a good example of something that was also done by Nellie Pitalou, who left our group to go to Lausanne, where she's a professor with Francois Prelong, another graduate of our program. So now we're beginning to look at this more in developmental terms. And you see that the neuroectoderm is the precursor here to these neurocrest cells, and that's where the pituitary stem cells come from, the hypothalamic stem cells that migrate in. And we're beginning to see the patterns emerge of developmental genes here with Cal and a variety of other ones. We also begin to see that the genes over here are those relating to the kisspeptin, but we're also beginning to see a bunch of genes in the middle that seem to have a footprint in both of these pathways. In addition, we began to see some patients that had 2 mutations. And 2 observations prompted this. The same mutation would occur within a family with different manifestations. The brother would be anosmic, the sister would be normosmic. In addition, other times when we would look at our mutation versus, say, French groups or Greek or Italian groups, we'd see the same mutation would give very different phenotypes. So we went and looked at this, and here's the first 400 of our cases and the first 8 genes that we had turned up. And the yellow shows that these are single mutations, and it shows you the incidence of them. Again, FGFR1 being the most common mutation. The blue shows homozygous or compound heterozygous mutations. But the red shows a second hit in another one of these genes, that's loss of function. So this is quite surprising. There aren't many genetic diseases proven to be oligogenic. And initially we didn't believe it, and in fact we went on and did almost 1,000 of these patients now, and you can see now with 14 genes, the oligogenicity is there. So this is the problem you have when you see families with penetrance. There's a second gene hit being involved in the interpretation of that most people don't know and understand. And it's been very interesting, and Ravi Balasubramanian and Jerry Sikiotis have now comparing this to the 91,000 databases of normals. So as a progress report, this looks not too bad on an NIH grant. You know, we're up to 35 genes over this period of time. But remember, what we've been talking about here is the power of Mendelian genetics. And they're severe and infrequent. We've shown you that oligogenicity occurs, and now common diseases happen with this. But they require lots of number of patients. And in fact, if you look at women with hypothalamic amenorrhea, this is the bridge to stress in the meeting. Nanette Santoro, when she was a fellow with me, described that all these patients have defects in endogenous GnRH secretion, either no pulses, low amplitude, low frequency, or developmental arrest. It's an AM/FM dial, if you think about it, amplitude frequency modulation. So we went and decided, is there a genetic basis for this? Not all women, but under stress, when they go to college, they lose weight, etc., they exercise heavily, some women get amenorrhea, and we looked at this and found out that many of these have heterozygous loss of function mutations. And none of the controls did, and none of the heavily exercising women did. There's also a surprising history of delayed puberty in these, in addition, a family history of hypothalamic amenorrhea that had been not documented. So the notion is that there's susceptibility genes that make you fragile in the environment, and it's a good example of genetics times environment. The ultimate one, of course, is we've been talking about rare diseases here. This could be Joe Goldstein's award of hypocholesterolemia, and this is normal cholesterols. And all we're doing is making transitions to those. And many of these rare genes are now being seen in common diseases. They map to the menopause and menarche. So complex trait genetics is going to offer a huge window into the future here. By the way, if you look at where's the power come from in basic biology versus human investigation, this is how Kalman syndrome was described by neuroanatomists. This is what won the Nobel Prize. All these red are basic research, except the observation where we showed GnRH in the humans. But look what's happened since then. This is all human genetics, brought to you 35 genes, and now with whole exomic sequencing, you can do this in scale. So we've been using phenotypic presentations, clinical syndromes, endogamy, that's what I just reviewed with you. We also have some structural variations, the broken chromosome and the FGFR1. We're also talking about logical candidates, but ultimately it's going to be whole genome sequencing in this cohort and populations that will, this is the tip of the iceberg, and it's going to be very complicated. And unless we understand the full genetic architecture of GnRH, we're not going to understand development, we're not going to understand pathophysiology, and we're going to be limited in designing therapeutic probes, such as kisspeptin, which is now in clinical trials. So with that, I'd leave you with the idea, this final slide is the most important one. Look at what's emerging here. You can see that in these genes, the crystopathies and the axonal guidance, the neural crest, this is the bioinformatic field relating to these genes. This is what happens in the neuroendocrine area. This is the field of genes here. And we've recently described a brand new syndrome of hypogonadism with a terrible CNS disorder. And in fact, if you look at that, there's a whole new field of these pituitary genes. So human investigation has been extraordinarily prismatic in this area. So what's missing here? None of Ron's transcription factors, no histone modifiers, Mark. I mean, and you know, hopefully that's going to turn up with whole exomic sequencing. So in summary, this is the contributions here of human genetics really prime the pump for basic scientists. It's a great opportunity. It's very enabling. There's significant oligogenicity here, and the convergence of Mendelian and complex trait genetics is going to be where a lot of this is going for candidate genes in the future. And next generation sequencing is going to empower this, and it's going to take off. Thank you very much for your attention.